Hello, Dr. Joel.
Good morning, Fabian. How are you?
Fine.
Yeah.
Nice to see you after 2 decades.
Yeah, after a long time, yes.
Yeah.
I'm really glad to have you connected through this webinar.
Hello, Dr. Joel.
Good morning, Fabian. How are you?
Fine.
Yeah.
Nice to see you after 2 decades.
Yeah, after a long time, yes.
Yeah.
I'm really glad to have you connected through this webinar.
A warm welcome to the Biotechnology webinar series on the theme Biotechnological Tools in Molecular Diagnosis. May I request you to be in a prayerful posture as we commit ourselves and this webinar series to God our Father.
Our ever-loving Father, the core of every reason, the source of truth. You who made the earth and the heavens, are the ultimate goal of every knowledge seekers, the infinite wisdom, the cause of all that is good. We are grateful for this opportunity to gather together as a community, despite the challenges we face each day. Yet you remained and gave us strength to carry on the responsibilities of bearing intelligence and using them for better purposes. We now humbly ask you to join us in our endeavor to explore the wonders of life in the world for knowledge's sake, that in whatever we learn today, we become more grounded to you, who is truth, who is love. We pray that you bless our speakers, the fount of your infinite knowledge, that they can share something worthwhile despite the limited time we have. They can channel all their boundless knowledge, that they may continue to persevere in the search and be deemed worthy to share what they learned from today's session. Sanctify us, O Lord, because we are worthy of it, but because because we believe in your love and mercy, that at the end of the day, we can take home something that's amazing and meaningful. This we ask and pray through Christ our Lord. Amen.
Good morning. Thank you for joining us for today's webinar hosted by the Technology of Loyola College. It's my pleasure to welcome every participant who have logged online and on YouTube. Thank you, Dr. Fabian, our resource person for the day. Thank you for readily accepting our invitation. Now I request Dr. J. Joel Jnanadas, Head Department of Plant Biology and Biotechnology, to felicitate the gathering.
Respected Dr. Fabian Davamani, the resource person for today's webinar and a very good friend. Dr. Sheila, the convener of this webinar, esteemed colleagues and participants, good morning to one and all. It gives me a great joy to greet you all and offer my felicitations on behalf of Loyola College Management and on behalf of the Department of Plant Biology and Biotechnology. I take this opportunity to welcome Dr. Fabian Davamani on behalf of the college and the department. Fabian is one of the illustrious alumni of our college. And not only that, he was working in the Department of Plant Biology and Biotechnology, and he was our own staff and a very popular teacher while he was at Loyola College. Welcome, Dr. Fabian, to this webinar, and I thank you for readily accepting this invitation. I also welcome all the participants to this webinar. The webinar series, a very interesting one, and today we are going to study about the molecular diagnostics. And molecular diagnostics is most sought after technique in the recent times, even during this COVID situation. It was one of the most important and vital methodology that was used. Molecular diagnostics is a collection of techniques which is used to analyze biological markers by applying molecular biology to medical testing. And it is one of the most significant discoveries in the field of molecular genomic technology. And today we will be having a lecture by Dr. Fabian on surface plasmon resonance-based diagnosis for biomarkers. As most of you may be aware, surface plasmon resonance, SPR, has become an important optical biosensing technology due to its real-time, label-free, and non-invasive nature. SPR is coming up as one of the most rapidly developing technique to aid detection of biomarkers and focuses on measuring even low concentration of the target analyte in any given test sample. This technique is one of the rapid technique and it facilitates ultra-sensitive detection of biological analytes with application in medical diagnostics, environmental monitoring, and also in agriculture. This morning we have a very fitting person who has done extensive research on molecular diagnostics. He is a product from our own university, IBMS University Davamani campus, and later on he had a lot of opportunities to work in some of the distinguished laboratories and become an expert in this field. I'm sure that today's proceedings will be very interesting and informative, and all the participants definitely will be having an enriching experience. I wish everyone all the best. Thank you.
Thank you, Dr. Joy, Head Department of Plant Biology and Biotechnology, for your warm words of welcome and for highlighting the importance of the series. We are privileged to have with us as resource person Dr. Fabian Amalraj, International Medical University, Kuala Lumpur, Malaysia. May I request Dr. Prem Kumar, Assistant Professor, PG Biotechnology, to introduce our resource person.
A very good morning to everyone present here. I extend a warm welcome to all of you for the Biotechnology web series, Biotechnology Tools in Molecular Diagnostics. I find immense pleasure in introducing our speaker of the day, Dr. Fabian Davamani Amalraj, Senior Lecturer, Biomedical Sciences, School of Health Sciences, International Medical University, Kuala Lumpur, Malaysia. We are really thankful to you, sir, for taking time off a busy schedule to do this webinar series. We hope that the students would be able to seek good notes and imbibe something from your talk, sir. Dr. Fabian was an adjunct faculty in the Department of Microbiology, Savita Dental College and Hospitals, Chennai, India. He was a Deputy Biosafety Officer in IMU. Then he was faculty representative IMU and a co-member of Emergency Response Team. Sir is an illustrious alumni of Loyola College. He served as a faculty at Loyola College, Department of Plant Biology and Biotechnology, and also at the Birla Institute of Technology and Sciences. He had completed his PhD from the Institute of Basic Medical Science, IPMS, University of Madras. He has worked on infections related to cornea from Real Ophthalmic Hospital and the Aravind Ophthalmic Hospital, Madurai. He has worked as a project at the National Institute in Immunology and postdoctoral researcher in molecular medicine and structural biology under the National Science Council, Taiwan. Sir has been awarded national level scholarship from Lady Tata Memorial Trust, Bombay, and Council of Scientific and Industrial Research, New Delhi. He has published several international peer-reviewed articles in various journals, and he is a member in the editorial board of journals related to microbiology. His research interests are in clinical microbiology, nano-antimicrobials, bacterial genomics, and molecular biology. Sir has won several Best Paper awards in international and national conferences and has worked on antibiotic resistance in clinical isolated and Antarctic bacteria. Now, I would like to request Sir to enlighten the students with his precious word as we begin this event. We welcome you, sir.
Am I audible?
Audible, sir.
Okay, thank you so much. It's a pleasure to come back to the place where I've studied and also worked for a quite some time. It's not a long time stint there before I moved out of Chennai. Yeah, it's been a pleasure always coming back to Loyola as well. And I don't call it as a lecture. Let me see it as a sharing. And thank you so much, Dr. Prem Kumar, for introducing me, and Dr. Sheila for her contacts. And we were over a lot of conversation for this to happen. And also, I thank her for all her efforts in bringing it up. Okay. So without any further ado, I'll try to get into the topic. So I'll try to share my screen. Is my screen visible for everyone?
Not yet, sir.
Not yet. Okay, let me share it again. So, the share screen is not working, so I'll redo it again. Okay. Is it visible now?
Yes,
Yes, sir.
Yes, sir. Yes.
Okay. Okay. Before I start, I'd like to tell you, like, I have worked with this SPR for some time, like few years, and had some hands-on experience. And my association with the structural biology lab, which is attached to a National Synchrotron Radiation Research Facility. This was an accidental thing where we were trying to sort out something in the lab. Then it gave a lot of insights to us. I'll also share something about the surface plasmon resonance and also some of my experience, how exciting this system was. And for example, I'll tell you one thing. This particular surface plasmon resonance is been, if you try to go back to your literature, you can search it even the past 30 years. The early 90s, they started it, but it's only with the genre of the biologists, not with the biologists, but with the physics people and a little bit to the chemistry people. And slowly, when the biologists got into it, then there's a boom. A lot of applications were found out, and it's really, really useful. I also feel fortunate to work in this technology when it began. Even before the automation, we used to do everything manually. Yes, that gives a lot of experience, and we could really appreciate the technique into that. And I'm a faculty at the International Medical University from Kuala Lumpur, Malaysia. So let's get into that topic. So let's try to understand in a simple terms. I'm trying to simplify and try to make you understand what is the surface plasmon resonance. Whenever we talk about plasmon or plasma, it's got to do with liquid crystals and all that, right? A quantum is a sum. So a sum of oscillation of certain liquids, right? So why does it oscillate? Because of some energy. This energy is going to be supplied by a light. So we have an optical source. Of course, when it is light, photons are involved, and the plasma will try to oscillate, right? And please feel free to ask questions if possible. So I can see only my screen, but not the other windows also. That's yeah. Okay. So probably we can have the questions at the end, right? So this is a plasmon, and what is a resonance? So resonance is a relatively large selective response, right? So the response is large, and it is also selective for an object or a system. Again, it vibrates. Again, it doesn't vibrate at random. It vibrates in a stepwise and or in a phase. So there may be definite phase. So when you try to give a signal which is harsh, the vibration will be more. If the signal, the vibration can be less for something which is very meek. All right. This is how we try to understand resonance as. Say, for example, there's a stream of water, undisturbed. You go undisturbed, then you see the waves. If you go disturb further, the waves will be bigger, something like a tsunami, right? The same thing we try to apply on a very microfluidic setup, right? So what we try to do, we try to vibrate by an externally applied oscillatory force. So this force will try to oscillate things. And what is surface plasmons? So these are surface electromagnetic waves. So the waves which are present on the surface, that will propagate in a directional, it doesn't go at random, which is parallel to a metal or a dielectric or a vacuum interface. So now we understand what is a plasmon, what is resonance, and we also have to do something with the surface. The surface necessarily have to be a metal. So what type of metal is being used here? We try to use a metal which is quite inert, not reactive with most other compounds. So gold is the best choice. We can still use silver, but still it has the oxidizing capacity. It's not being used. So a very thin layer of gold can be used to coat. Even with 1 gram of gold, we can coat several hundred chips with that. All right. So this is something about surface plasmon. Basically, we need a surface with a metal, and this metal is going to be gold. And we need a plasma, means some liquid on top, and we need a light energy source so that we can track the direction of the light, and it's going to be deflected. We'll also try to track where it is deflected. Then the whole concept of surface plasmon resonance will be very clear. All right. So this is a technique which doesn't need any labeling. Say, for example, your ELISA or any other technique, we used to have a label. We have to use a fluorescent label or anything which can give a color and things like that. But here, when two things bind, you don't have to use any other label. I'll try to burden the system. Just when it binds, we can exactly see what exactly happens in the binding of these molecules. So it is based on molecular interaction, meaning that I cannot take two cells and try to react and try to get something out of a surface plasmon resonance. It's not possible. So there's also a limit, right? I cannot take a huge molecule and try to put it inside this particular system, right? So these are molecular interaction. Maybe a protein or even a virus can get in, but a whole cell, you can immobilize it, but still the signal can be quite weak. Right. So what exactly happens here? We have an incident light. Earlier we used only lasers, but now with the LEDs, which are very powerful, which can give different colors of light and with a different range. So with a single beam source, we can get the light, the polarizer light. So basically, you try to have a gold film, which is about 50 nanometer, 40 to 50 nanometer is fine. And then you have a prism, then you send the light. On top of that, you are going to have a liquid. In this liquid, you can immobilize something. Then whatever is immobilized, it will try to take the reading, and that can be measured. If something else tries to bind to the immobilized protein or whatever it is, then there's a reflected light. Then there's a little bit of change, right? Here they are given an area of about 500 nanometer. Now we can also extend it up to 1000 nanometer or 1 micrometer. So this is a depth in which we can measure things which is on the surface, right? So now we understand that the surface plasmon resonance is something which is happening on the surface of a metal plate, right? That is very thin, and we try to excite it with a light source, and it's going to deflect. So when it creates a plasmon or a resonance, so based on the type of molecules which are there, you have one molecule, it will be different. If it binds with another molecule, then it becomes a little bit heavy. Then there will be a definite reflection of the light, right? So this is exactly what we try to do in a surface plasmon resonance. Okay, how does this things look like? So here I have shown different type of chips, right? So this is a chip which is used in the surface plasmon resonance, right? And there are many commercial companies which will try to make this chip. And if somebody knows the principle very well, then nowadays we can also apply this to a mobile phone. So the technology is in place, but mobile phone has got its own limitations. We can detect 1 or 2 parameters by using a mobile phone. By using a real chip, you can go up to 36 parameters can be measured at a single point of time. So why I'm telling all this is like, again, you need very, very low quantity of your sample. Say, for example, if you have 300 microliter of liquid, which has got anything that is more than enough, and by using some of the devices, we can also recover 200 microliter back for some other test. So this is the amount of fluid which is required here. Again, every time, whenever we try to use a liquid, we have to use with utmost caution. We cannot take any solid particles inside, which is few microns in length. Then it will try to clog all the system. So these are microfluidic devices. We have to keep it in mind. And it's not necessary every time you have to use a filter to filter it out, 0.22 and all that. No, you can just centrifuge in a high speed, like 10 to 12,000 RPM for 15 minutes, take out the supernatant, and you can start loading into it without disturbing the tubes, right? So there are ways in which how you can handle the fluidic system, microfluidic system, but we have to keep it in caution in a subconscious mind that no solid particle should get into the microfluidic system, right? If it gets into it, it's going to be blocked. The whole system has to be cleaned, or sometimes they go even useless. All right, okay, next. So whenever we talk about the chip, what is this coated with? We know that it is coated with gold, and I cannot take and attach everything to the gold, right? So I have to have a matrix. So there are different type of matrix which are commercially available, and we can also treat the surface of gold and try to attach whatever matrix we have. This is a flexibility of the system. So earlier people use a cellulose, methyl cellulose, or a nickel NTA, or a heparin or gold, whatever it is, right? So we can attach anything, even oligos can be attached, like your DNA molecules and things like that can be attached to the chip. And the matrix can be anything. So one thing, we have to treat the surface of gold, and then we try to attach to it. There is a chemistry behind it, or we also have some commercial kits. If you're not very familiar, you can buy the commercial kits and try to activate the surface and try to bind whatever protein you want to catch the protein or anything of this. Matrix is the one which is going to hold your protein or the capture molecule, right? And this can be very, very varied. You can attach a DNA, you can attach a protein, you can attach a glycolipid, or you can attach a lipid alone. Anything can be attached. Okay. So we have, this is about the matrix, and this slide is about 15 years old. So even 15 years ago, or 50 or 20 years ago, people had this technology commercially available, right? So this particular thing, I'm tracing back over 30 years. So people had different chip. So you see like a heparin chip. It's basically, earlier we had only one company, which was like a monopoly. It's called as a Biocor. Now it's with another company. So this particular company, which started the SPR machines and other things, they were the first to come out with most commercial kits and other things. So even in the 1993, means like they should have come out even with the 1990s, so that it's published in 93. So this is a 30-year story where people has been using this particular technique. Basically, they're testing only with lipids, certain chemicals, certain molecules and other things. And slowly it came into application after 10 years. So in the past 20 years, a lot of work has been done. The past 10 years has been the really fastest period where a lot of modification has happened, or improvements or simplification happened with the SPR surface chemistry. All right. Okay, now I have put 3 type of flow cell. So you might wonder how this whole thing is going to work. For example, I take the first flow cell, CD4, D3, D3 and 4 and all. So this particular thing has a flow cell named as flow cell 1, 2, 3 and 4. Meaning that I can put the sample from one side, it can go through 4 different flow cell and come out. So with each and every flow cell, it will try to take the reading. The first cell, we always have it as a control. We don't put anything. The second cell can also be another control. And the third and fourth cell, we can use it for diagnosis. Right. Usually, people try to use the first cell control, and second, third, fourth, you can still use it for diagnosis. So it is mandatory that you should have at least 1 or 2 controls whenever we try to do this kind of experiments. And the data has to be at least duplicated or triplicated, because you can run the samples again, since we can recover it if it is a very precious sample, or we can do multiple injections. Here we talk something like injections, because these are all fluids. So we try to inject the fluid, then it goes through the flow cell. As it goes through the flow cell, when there is a binding, we can find out by using the principle of surface plasmon resonance. Okay, this is one type. Another flow type, I can have 6 different sample inlet. Right. The first case, there is only one sample inlet which will go to different flow cells. So if I have 6 different tools, here I have about 8 different samples. So this is usually done for the molecular diagnosis. So when I have a lot of samples coming in to the lab, right, I can have 8, even I can have up to 36, where 2 can be controls, and I can run even 34 samples at one shot. So all this can be automated, and what I required is only about 300 microliter of the sample. Say for example, you can take even the saliva. You don't have to take really the blood or the swab from a patient and try to dilute it, take the fluid and just run it through that. So you can detect multiple parameters even with a single sample. So we have a detection spots. When you see these detection spots, we can have more than one parameter in the detection spots, right? One is for control. We can have detect more things, right? So the flow cell can also be designed in a way which is flexible. Right, I can use multiple injection, or I can use a single injection and try to do multiple parameters, or multiple parameters with multiple injection. Anything is possible, and these things are done by lithography. Again, these days we have computers. Just feed in whatever you want. You get the lithography printed out, and the flow cell is ready. Right, and people can also use some improvisation into the SPR flow cells. They can add, they can use magnetism, they can use fluorescent labels, and they can use a nanoparticle, and all that can be incorporated to even enhance the detection limit, right? So in the later slides, we'll try to see how far is the detection limit of these kind of systems. So this is about the surface plasmon resonance, where this is a microfluidic system where there is a flow cell. The flow cell is basically where the surface plasmon resonance happens. So you have all the gold coating and other things will be only the flow cell. You already coat your capture molecule, and then it will try to capture whatever ligand you could try to put in your samples. Then that will be red. You don't have to add another antibody and other things. Just the binding is more than enough for you to detect it. Right. Next. So how does these machines look like? So there are several machines which are available in the market. So based on your budget, you can go for a machine. If you are not able to buy a machine, you can also build your own machine by using certain optics, laser or LED, and even with a space like what you see here, there are also manual methods. You can always build up your own surface plasmon resonance. The only thing, if you have a chip and a light source and a detector, you can build your own system. But of course, yes, it needs the expertise from the optical side as well as with a physics expert, so they can do it. And these techniques have been very, very well developed over the past 30 years, and it's very rapid, and it's now the size of these machines are also become very, very, very small. Yes. Yeah. Okay. I'm telling you, like, this particular machine can also be used with a mobile phone. Here's an example. So what they try to do, they try to develop this particular method by using a very small capillary, right? Within that, they have a flow cell, and this one can detect only one parameter at one time, right? So for example, you try to do your controls and take the values, and then you run your samples, then you try to collect the data. Once you try to collect the data, you'll be able to do the diagnosis very fast. For example, now we have a current outbreak, right? Not everybody can buy the huge machines. Probably these kind of techniques can be very, very handy. But only recently people are getting into the real-time PCR and other things, which takes a lot of time, and also it is quite expensive technique. It's also a quite expensive technique, where it's also needs a lot of skills. But here, if everything is set by the technician, so the by the scientist, the technician can run the sample, immediately look into the data and try to see whether it's a positive or negative. They can also get more parameters, what we'll try to discuss later in the later slides. Okay. Now we try to come to the topic proper. So what is surface plasmon resonance in a crux? Say, for example, I might have a coating in here, right? Then I'll try to put the sample, then it will first try to get associated. So you can see the exact curve when the molecule get associated, right? Means that there is a binding happening. As the binding happens, you can see the curve which is coming in. So this is what is a kinetics. Again, this can be dependent on the concentration as well, right? So another thing, what we try to do, we try to disassociate the bound ligand, meaning that the ligand is bound with the capture molecule. So I use another buffer so that the ligand leaves. If the ligand is not going to leave easily, then we can tell it's a very strong binding, right? If the ligand is going to go immediately after I put it, then it's a weak binding. Even the binding kinetics can be worked out, right? So this has got a lot of advantages. So once we use a binding buffer, then dissociation buffer, then the regeneration buffer is a strong thing which will try to clear totally all the things which has bound to the surface molecule. And now the chip is ready for the next test. So here it's not an ELISA or other test where when you try to do one experiment, you throw away the things. No, here we can reuse the same chip for many times, right? It also depends on the stability of the proteins and other things. Once we can do a test run, then at least like 40 or even 50 or hundreds of times these chips can be used, because this is an online monitoring. As the binding happens, you can see the data coming out, right? So you can see whether with regeneration everything is moved out and the chip is ready or something is blocking and other things. You'll be able to do a lot of QC, even to monitor what exactly happens in a real time. All right. So this is my experiment. So I took a protein and a mutant. The way it tries to bind, there was a difference, right? So when we put the mutant, it totally stops cancer. When we put the right protein, cancer can go on. And this is also one of the biomarker, hepatomer-derived growth factor. But we took only the first N-terminal 100 amino acid and took the protein and did all this kind of experiments. We found the SPR to be very, very sensitive with the binding kinetics. Because the mutant doesn't bind well, so we can see that association slightly different from the real protein, right? Again, when we use a peptide, because the molecule is very small, it cannot be compared with that. So I have to expand only the lower age regions. Then you can see how good is the binding with the peptide and other things. Basically, the peptide didn't bind to it, even though the peptide had the active site. So it needs the other components from the protein to bind to make it happen, right? So this was very clear. We also published this data earlier. So, yeah. Okay, but how do I do it? Whenever we try to do it, I said we have to do it with a different concentration gradient, right? So if you try to look into that, I started with 10 nanomolar till 100 nanomolar, right? So I use the protein, and the volume is 210 microliter, right? So I try to do 2 times injection for each and everything. It has been repeated, and this is how we try to set up. I can set it up and just leave it. Once I set it up and put it in the computer, it will take the sample automatically and try to inject one after the other. Then I can study the kinetics, right? So this is what happened. So I'll try to inject the sample based on different concentration, and I can see the association, which is very nice. And the dissociation does not happen immediately, meaning that the protein bond very strongly to the capture molecules, right? The same thing we also done with the mutant. When we found the mutant binding was not as good as a real protein, right? And the dissociation constant is also slightly different from the real protein. And when we did with the peptide, there was no association. So this is very clear. So this is also a biomarker. So even different type of biomarker, you can see the difference even within the same biomarker. The difference between the mutant and the real protein is only 1 amino acid difference at the 96th amino acid position, right? So this is such a sensitive method where we can see even a single amino acid mutation, how the kinetics is going to be changed. No other method could tell us so much of details like what we could see from a surface plasmon resonance. Okay, so this is how I try to do the injection. So I can see if there is a difference between the 2 injections, there will be some slight difference because this is a very, very, very sensitive. So based on this difference, I can still optimize the kinetics. I can go to the kinetics and try to see the difference and do a mathematical optimization and try to even optimize these 2 and try to get even the best value with the highest accuracy. That's what we call finicky, means there is no change. So we try to get those kind of very accurate data with this kind of injections. So everything is back traceable. Even though you finish your experiment, you can go back to the machine and try to see how your injections are. If the 2 injections, if there is a difference, how much is the difference? And what is the difference between the absorbance, what we've tried to find out on the SPR. And this can be calculated back, and we try to know all the details, right? And the whole thing, it takes less than 10 minutes for each and every sample, right? If you run a real-time PCR, it takes, for diagnosis, it takes about 3 hours, or at least like 1.5 to 2 hours. Okay, I said people have already gone into this particular technique and enhanced it. So we also have something called as surface enhanced Raman. So we can incorporate the Raman effect into the SPR, and you can get the enhancement 10 to the power minus 14. So this is a fraction and fraction of things where the sensitivity is going to be increased. 10 power 14 is way far beyond sub picomolar range. Right? Again, there are a lot of, we also can enhance with infrared, right? These were basically done with different molecules. When they said that we can not only use SPR, I can also enhance this by other possible technique, then my sensitivity can be really, really good, right? Like that, they even use a fluorescent method where they were able to enhance it by 100 times and so on. All these things is, if you try to trace back, it is about like 40 years ago, 1981. So this technique was used only by certain physicists and the people who are involved with microfluidics, so the chemists and other things. Now I tell you, like, this is a 40-year-old technique, right? So 1980, 1981, already people had this kind of data in. Yes. So now it's better. Yeah. So what is the advantage and potential artifacts? So the advantage is this is a real-time system, right? As you try to run that experiment, even before the experiment is over, you can see, monitor each and every step which is happening in the computer. And you don't need any extra labeling, right? And no external labeling, no internal labeling is involved. Very small volume sample, 300 microliters more than enough. And sub-micro and sub-nano range can be achieved very easily. And it is highly versatile, means it is very flexible with different kind of system. I can use it for a lipid, I can use it for a DNA, I can use it for RNA, I can use it for a microRNA, anything, right? So again, there are certain limits. So we have to look into the immobilization effect. Whatever you try to immobilize should be stable. And if the immobilization effect is not good, if it's going to run away, then it cannot be used. There can also be some steric hindrance because of the forces, the free energy and other things. And the non-specific bindings can also happen, but that will try to literally make a mess with your data, whatever you try to get. So all these things has to be prior worked out before we try to go into the SPR. And the mass transportation limitation is like we cannot take bulk molecules and put it inside. And these are the limitations, but still it's a very useful technique, right? Again, we have to do some very careful design of the control experiments. Without control, you cannot just go ahead with it. And of course, data analysis nowadays is very simplified. Earlier, we have to apply the formula and calculate. Yes, all these things have overcome very well in the recent past. Okay, sorry. So what are the, now let me get into that. Now we know what is the technique, how it is used, how good it is, how it can also be enhanced and all that. So now we talk about biomarkers for surface plasmon resonance. So whenever we talk about surface plasmon resonance, the sample is only a liquid biopsy, liquid. So we have a lot of circulating biomarkers in peripheral blood. You don't have to be very invasive, meaning like you don't have to take a cut a sample and try to extract things and know, right? For example, microRNAs, exosomes, circulating tumor DNA is there, circulating tumor cells can be there. We can extract that and do it. And of course, proteins, most of the things, whatever we try to use is proteins and other things. So anything in this list can be used for surface plasmon resonance. Okay, here I'm giving you an example. If you try to look into the first in the list, people had taken miRNA and it's the bladder cancer cell. They have gone up to zeptomolar, which is 10 power minus 21, right? Imagine micro is 10 power 3, then nano is 10 power 9. So it goes on like that. So 10 power 21. So even this is the type of sensitivity which we can get it with SPR, right? So the detection limit is fantastic, and they have used about 140 megahertz of energy. That is a type of energy which is given through the light source, and they can get it up to a zeptomolar. Now, probably this might be a new term for some of the biologists. Yes. So this is a kind of extent we can go with the sensitivity, right? Another thing is a femtomolar. This is also sub-nanomolar. This is 10 power minus 15. And this is all, what I'm talking is all published data. The zeptomolar data was published in 2019. Earlier, the femtomolar was the best. It was done in 2014 itself, right? So that also they check with the miRNA. The biological specimen is just a plasma. Plasma is just a blood. You collect the blood, you clot the blood, whatever remains on top is a plasma. Take that and inject into the system. This is the kind of measurements what you can get it for these kind of molecules, which are very, very low in number, right? And they can be detected by these kind of very sensitive techniques without any labeling. That's another advantage, right? So we also have attomolar, which is slightly above the femtomolar range. It is 10 power 18, right? So this is also in the same year that they took the exosome. They took the exosome, which is a biomarker or a human ovarian cancer cell line, and they were able to detect it at 10 power minus 18 molar, right? That's an attomolar range. Again, of course, like some of the mutations in the gene were also be able to detect it at a very, very low concentration of about 10 power minus 9. That's a nanogram concentration of the DNA can be measured. And also in breast cancer, people has gone up to picogram levels for detections. And also in multiple myeloma, which is a very, very, very difficult thing in cancer diagnosis. And people has gone for picomolar or range of sensitivity, right? So if you try to look into this data, all this published data which has used surface plasmon resonance, right? And this data is something very authentic. Again, this data is very real because like as it runs, you get the data and nothing can be done, right? So now it has become a very affordable technique, and it's got no ambiguity, just the data. We'll have to believe it. There's nothing in between that. So these data are very, very reliable and very, very, now they have found a lot of applications. Okay, so there are also a lot of examples, right? So whenever we talk about biomarkers, the best way, go and search your literature properly, or you can also develop your own biomarkers. For example, there is a talk tomorrow which is taking the DNA part. Even the DNA, you can make an array of 96 well array. And now the recent one, you can take about 384 array in a SPR and try to detect so many molecules based on RNA biomarkers, right? So the technique is always like moving forward in a very fast pace in the recent years. All right. Again, how about viral diagnosis? They have done it with the HIV by using a CXCR4 receptor. That binding, they can see the concentration dependent binding, where they can clearly see these bindings are very, very specific. You can suspect the people with this viral disease, run it through an SPR for 10 minutes, then we can do a HIV diagnosis. That's fantastic. So is it relevant to what is happening recently? Right. Now we have the SARS. The question in your mind, I know, yes, can this technique come in for SARS-CoV-2 diagnosis? The answer is yes. And people has already done, and they published this in 2020 December, right? So people are able to take the SARS-CoV, and here they use a nanoparticle. So whenever you try to use a nanoparticle, you always have a single surface, and it's a nanoparticle. It can be a round nanoparticle. Now the surface is also a little bit enhanced because the nanoparticle can be also a metal. Then the resonance can still be better, so the sensitivity can still be better, right? So by enhancing this kind of SPR technique, so we can still go for a better diagnosis, right? And this is a published paper. I'm just giving it to you. And another paper also on surface plasmon resonance biosensor for SARS-CoV-2. This is also published in the 2020 September. Before that, so they have compared all the technique from CT to PCR for ELISA, chemiluminescent, and lateral flow strips. Everything they tested, and they said the biosensor is going to be the most fast, ultra sensitive, and they can do it on site, and it is the lowest cost. So when we try, so this is the recent thing what we are getting from the researchers, in also from the field experts. So this is a fastest technique, and the sensitivity is very, very good, and you can take it anywhere on site, and the lowest cost. So what else you need? No complications. Everything is. So this is a technology which is also planned for futuristic. Anything coming new, you can still apply these kind of techniques. Okay. Again, talking about biomarkers, we always have a confusion. How do I get all the biomarkers? Don't break your head. Don't have a head spin. There are different type of websites where you can go to the database straight away. So what I've displayed is a marker DB from California University. So they have different biomarkers for different conditions. It also includes biomarkers for infectious disease, for disorder and other diseases like cancer, and for anything and everything. They have a database, not only one. So we have several databases which you can go and search for the biomarkers. If you're really getting into a disease, you can get the database, see what has been done. You can also go to the other sources and try to find out, even from the microarray data, what all the things which are reflected. So you can create your own kit by using an SPR and go for a quick diagnosis. All right. Okay. Biomarkers in general, again, to just give you a crux. So we have the tumor circulating DNA, and you have the cells, you have the, we can detect even point mutations, we can detect the exosomes, we can detect proteins, we can detect even DNA methylation, right? We can detect microRNAs and interfering RNAs and anything which is possible. So this is a solid technology based on liquid samples. No solids, no aggregates, yes. So the technology is wonderful, it is applicable, it has been proven over 40 years of time, and it has lot of improvements has been done, lot of new things have come. Now it's very easy for you to use, right? If early days, if you want to use SPR, it was something which is not reachable. It was very complicated. All the complication has been rectified. Anybody can use it. So I would recommend you all think of this technology if you're going in for research or if you're going in for a diagnostic kit and other things, right? So here, I think I've just taken my time. So about 40 minutes. So SPR is a label-free technology for studying biomolecular interaction in real time, right? Second, this is a diversity of immobilization. There are different type of immobilization methods available. Anything you want, you can always find a matrix to attach it, right? So the surface chemistry has been thoroughly worked out. You just go through literature, follow the method, develop your own. Or if you don't have that kind of time, then you can buy a commercial kit and still do it. Again, critical awareness and potential limitations. This has to be kept in mind. Of course, you have to have knowledge of your protein chemistry if you're working with proteins or liquid. If you're working with a lipid, if you're working with DNA and other things, because you should know how to handle these molecules in microfluidics. Of course, most of the people have experience in all that, and this is going to be the technology which can bring, which opens up the Pandora box in diagnosis. So with this, I'll stop here. I think I've exactly taken about like 40 to 45 minutes, and I'll be glad to answer your questions, whatever you have. Thank you.
Thank you, Dr. Fabian, for your very intellectually stimulating talk on surface plasmon resonance-based diagnosis for biomarkers. Thank you for highlighting its working, principle, and current applications of the SPR systems. Within an hour, you have taken us on a very interesting journey that captured the development of surface plasmon resonance over the last 4 decades. The sensitivity of detection without labeling to the level of zeptomolar concentrations is highly commendable. And you also highlighted the most recent application of detection of the novel coronavirus. We also appreciate how you brought out that this tool is an example of how biotechnology unites scientific disciplines like physics, chemistry, and biology. Thank you, sir, for sharing your wealth of knowledge with us. Your talk has motivated our students to make surface plasmon resonance themselves possible and contribute to improving healthcare. So now the floor is open for questions. We do have a few questions already, sir, so I can start with the ones on the chat box.
Yes.
So the first one is, please could you tell us how controls in surface plasmon resonance?
Okay. Say for example, you have to keep one well without coating anything. so that if you have something in your buffers and other things, then you should not get any signal. If you get any signal in an unquoted thing, then your buffers, you have to check it again. Again, it will also tell if there is any particulate things which is running around in your system. Right. The second thing, you have to quote only the, say for example, you have the matrix. Matrix alone, you should have another one more control. Then only you can feel that if there is any non-specific thing which can be bound with the matrix, or you can just quote with some BSA, right? That will serve that if there is anything non-binding, non-specific binding happens, then the third and fourth or even up to 36 flow cell can be available. So then you can put different parameters just to see if there is something wrong with the system and fluidics. Then once you do an experiment, all the samples should not go waste. Again, there's another possibility. You can just run the buffer through all the flow cell and try to see to that, okay, everything is clear, even if something is blocking somewhere. So even after your run, you can still go back and check how much of things has been injected and all that. So this probably I started with a very manual where automated injection was not possible. We have to take the micropipette and try to inject things into the flow cell. Then it goes in, then all that. So when automation came, we found it to be very, very useful. Of course, now there is no more manual machines available, but if you try to create your own SPR, you can still do manually. It can be done at 1/10 of the cost of a commercial machine, whatever is available. But you have to work with some physics guys and chemists to do something on your own. Yeah, controls are very essential. Yes.
Thank you, sir. Another question is how to perform multipolar analysis in a resonant metamaterial structure.
Okay, again, you have to have more controls. So when you add in any material, so that will give a reading. Say for example, you are adding a nanoparticle, or if you are adding a metal nanoparticle, it will be different. Because the surface plasmon resonance, the resonating thing for each and every molecule, even if it's a protein, it's different. Even within the protein, if there's a one mutation, it can be different. Even if it's a metal, it's different, right? Say for example, nowadays people are enhancing with a nanometal on top. So this metal can have a higher surface area than just by a protein which is just exposed. Then you have to increase the depth of detection from the surface. Earlier I said only 500 nanometer. Now we can go up to like 2000 nanometer techniques is available, right? So you have to opt for those kind of options if you try to go in for more how do I tell, improvise things. Then of course, yes, you have to have more energy. You have to do all the control run. Yes, things has to be optimized based on the chemistry, whatever you're having in a matrix and their molecules. Then of course it can be done. The only thing you have to sit down and titrate it and try to see where it works. Then you have to get to that point and try to work on that.
Thank you, sir. Another question is it possible to detect non-culturable bacteria?
Yeah, we can detect it. Again, the probe is a problem. Because nowadays they are doing the 16S RNA. So you can have the 16S probe and detect even like 96 or even 384 at one shot. So you can get all the probes, and again, you can, because I'm not talking much about the oligo, you have another talk. So you can take those oligos, which will be spoken to you the next day, and put it on an SPR. Definitely you can, up to 384 is possible. So you can go ahead with all this kind of, instead of using the Illumina, you can still go in for this surface plasmon resonance. But it is still in development by many universities are still working. Probably in the nearest future, I think it will be commercialized as well, because it works cheaper than the Illumina.
Thank you, sir. Another question is, what are the limitations of SPR?
Limitations is the binding. So you have to have some specific binding. If it is a non-specific binding, probably again you have to go back to your certain classical techniques. So if you have, again, otherwise you have to develop a system which where we can have an enhancement by using a nanometal or a nanoparticle attached to it, so that it specifically captures. Right. So there are different types of SPR. I'm not getting into the details of that in this lecture. So you can choose that option which will suit you. Definitely there is an answer. Right. So that will be the suggestions from me.
Thank you, sir. Another question is, could you give us examples of matrix used in SPR?
Okay, several matrix. You can use any matrix. Okay, the best thing is that you can also create the surface of the gold and make, you can do a surface chemistry based on certain acids and base. So the surface is exposed, then you can attach anything you want, right? So it's exactly doing like it's like an ELISA. You try to coat, then you exactly like that. You have an aldehyde chemistry. You have different chemistry which is available. If you want to coat an amino acid, the chemistry is different. If you want to coat a lipid, the chemistry is different. If you want to coat a DNA, the surface chemistry is different, right? The surface chemistry alone can be another lecture, right? So if you can work on the surface, but literature is already available, very clear literature. So see to that what molecules you have, select the right chemistry, and buy the kits or buy the reagents, and you can do it on your own. So this is how it is.
Thank you, sir. We'll just take two more last questions. Is that okay with you, sir?
Yeah, sure, fine.
So one is, how does it relate to the Raman effect? And the last one is, can Nympha virus be found using in SPR?
Okay, I'll go one by one. First is the Raman. So the Raman also work on deflection. The peak will slightly shift, right? When you have a shift in the SPR also, plus Raman, right? A very minute things. The both shift put together, you can see a very big difference. Say for example, if you are having a nanomolar, it can go to a picomolar sensitivity by combining these two techniques, and it can also be done on the SPR surface itself, right? So there are, but the SPR has to be modified in such a way to incorporate the Raman. So the commercial things we cannot do it. So you have to develop something like a benchtop SPR. On top of that, you have to also incorporate the Raman, and then put your samples inside. Then definitely you can get the range. Okay, the next question is Nipah virus. Yes, any virus is possible. So you try to get the surface protein, which is very unique for this virus, and get the complementary thing, or you can take the DNA, take the lysate. It's easy to diagnose it in that way, and or you can take anything the virus can bind to. Some of the virus sample, they bind to certain receptors. You can take the receptor, and for example, some toll receptors are involved. Certain things, sometimes the cell surface heparin sulfate is involved. So you can take all those things and try to do coating and capture the virus. Or simple thing, use an antibody straight. Even though you use an antibody on a single chip, you can completely strip it. Again, you can redo the chemistry. Say for example, you run for about 100 tests. Okay, remove the antibody and recoat it. You can use the same chip. But the commercial people will tell you cannot use. But if your chemistry is good, you can still use it. But so these are kind of, we'll be breaking the trade secrets by using a strong chemistry. So we can even use the chips multiple times. Just reactivate the surface and do the coating again. I can still run through another set of experiments and things. So this is a kind of flexibility we will have with the systems. You can use it for any virus.
Thank you so much, sir. You really answered all the questions. Appreciate it. May I now request Ms. Ariba Khan to propose the vote of thanks.
Good afternoon to all. As all good things end in life, so here is this seminar for today. On behalf of Loyola College, I take this opportunity to propose vote of thanks to those who have directly and indirectly contributed to this seminar on SPR technology. I thank our honorable chief guest, Dr. Fabian, sir. We are really enlightened with your knowledge, and thank you for sharing your knowledge on this SPR technology. I'd like to thank our HOD, Dr. Joel Nyandas, for his enthusiastic support. A special thanks to the organizing committee, teaching and non-teaching staff for the support and coordination. Our heartfelt thank to the students for their participation in the seminar. With these warm words and kind message, we move to the end of today's seminar. Thank you everyone for your patience, and thank you to everyone. Thank you, ma'am.
Thank you, Arita. So this brings us to the close of day 1 of the Biotechnological Tools in Molecular Diagnosis webinar series. We look forward to meeting you at tomorrow's session, which will commence at 11 o'clock IST. The feedback form will be posted at the end of tomorrow's session. And on behalf of the PG Biotechnology Loyola College Autonomous Chennai, we thank each of you for your active participation and we wish you a pleasant day. Thank you so much.
Thank you so much.